Let's do this problem. Capital T, dominant allele, is tall. And the recessive allele is short. Okay, so we were given that. Or dwarf. If you have a tall plant, we don't know. So let's say tall plant cross with test cross. Test cross is the homozygous recessive. Okay, so you do test cross to figure out the genotype of the unknown. How can it be determined if the plant is heterozygous or homozygous tall? So just because it's tall, we don't know the genotype is heterozygous or homozygous dominant. We don't know that. Okay, so that's why we do test cross. If it was homozygous dominant crossed with recessive homozygous recessive, then when you do Punnett square, 100% all the next generation, F1 generation will be tall because their genotype will be heterozygous. This will be tall. 100% will be tall. However, let me do a different color. If the parent unknown was heterozygous to start with, if you cross with this, okay, when you do Punnett square, it's going to be half or 50%, it's going to be tall with this genotype, and then half it will be dwarf. Okay, so half will be tall, and half will be so 50%. I'm just gonna do one half. Half will be what is it? Dwarf. Okay, so by doing test cross and looking at the frequency of the next generation, you could tell whether the unknown was heterozygous. If the children were like, not children, plants, the next generation would be half tall, half dwarf. Oh, then the unknown must be heterozygous. Or all the next generation is tall, then the unknown must have been homozygous dominant. That's how you figure out whether it was heterozygous tall or homozygous tall.